All right, so next we have Lydia. So Lydia is here at Allen Institute, and she's been involved in the um, BICAN. So she's been involved in the BICAN, and she's going to tell us about like the Common Coordinate Framework, which is becoming a quintessential like tool for all of us. Because when we bring data, we want to be able to talk about some reference points. So this is going to help us to discuss it.
Thank you. Oh, I'm just talking into the mic. Yes. All right. Hi, I'm Lydia Ng. I am at the Allen Institute for Brain Science. I'm an investigator in the data technology team. And today, we're talking about standards and CCF. My talk, unlike other people, is like a Sunday stroll. We're going to look at some history and what people are doing now and sort of the road ahead as well. I named this a 10-year anniversary, but I was putting this talk together. There was a previous 10 years, and if you were getting lunch, there were those plaque cards, I guess, on the wall. On the other side of it, there were some dates. And basically, the journey of the CCF really, matches onto all the different projects the Allen Institute has since starting at 2003. So our very first project at the Allen Institute, if everybody remember, was trying to make a complete gene expression atlas of the mouse brain. So that's ISH data, every probe, so 20 plus K of data. And at that time, in 2004, I guess the gold standard or standard practice, if you're looking at data, is your book atlas, and then you have your picture next to you pick your best atlas plate, and then you have to do some inference. But because we're about to generate such a large data, we needed a way to sort of do things more computationally. Hence, probably the startup of the informatics team at the Allen Institute as well. So our very first Common Coordinate Framework actually came out in 2005. It is from a single mouse, the one that's same as the Allen Reference Atlas. At that point in time, we had it at 200 microns. It matched our sampling density for our ISH, and there was only about 200 structures because each of these plates were hand-drawn. And when we picked these 200 structure, they were the bigger structure, and we sort of went back and forth with the anatomist to say, hey, something is a bit off here. So there was a lot of cleanup of those 200 structures. And that sufficed because because the data was sparse, it was enough to get a good look at, do some searches on the data. As we enter 2011, was the beginning of when we did the connectivity atlas. And at that point, tissue site data came to being, and it was expensive. It took a day to image a brain. And what was unique about it was that it came with 3D data. And so the idea is that you really do need something computational first to find out where the signal is, and then to compare each data set. So a sort of a sparse atlas wasn't going to be sufficient because we wanted to, where the projection or connectivity went to small parcellation of the brains. So that was our first redo of the atlas in 2011, in which we go, okay, we're just going to take every drawing that was done in the book atlas and stick it into the CCF. And then we upped the sort of the sampling to 100 microns, and that was really the first version of the connectivity atlas. All the results from that paper was with this version of the CCF. But this this connectivity atlas actually gave us a very unique opportunity because of the tissue site data. At this time, this was probably the largest collection of high-resolution 3D data at that time. And this gave us an opportunity, well, let's make a reference atlas out of that. We started down that track anyway because registering tissue site to the NISA was difficult, so we started making average templates. And with this average templates, we were looking at very sort of clean registration. And then at that point in time, Hong Kui Zhang said to let's boot up a atlas making activity with the first version released in 2015. Ending in 2017 because it's multiple years. So making a new atlas, it takes a village, it takes many years. So at that time, when we first started, there were about 2,000 mice that we had imaged, so all of it came to be used in atlas. We have way more data than that now. We took it at a high resolution of 10 micron. 10 micron, wasn't single cell data, but sufficient to see a lot of details. And then a multi-year annotation project was kicked off, supervised by Julie Harris, led by Chuanchen Wang and Songlin Ding. And Yang Li here did a lot of the heavy lifting of creating this average template. At the end of that, we had like 43 isocortical areas because at Allen, we're very interested in cortical analysis. And of 329 subcortical areas were annotated as well. And what we wanted to do was make sure this data, this product was open access. Anybody can download it as well. All right, so again, it was because of this data set, the Allen Connectivity Atlas, we had access to that many specimens. I think if we just said we were just going to sacrifice 2,000 mice just to make an atlas, I don't think that would have been a feasible attempt. But because of this data set, we could reuse it. And because injections were in different places, by the time you averaged out that many animals, the injection site wasn't going to have a big effect on the average template. Okay, so we took our 1,675 mice and we created a average template, which was both an average intensity and also an average shape. So each time you do registration, not only do you take the average, you take the average transform and back out the average transform because your first version of it may have some weird kink, whatever you start with. And then so as each data set registers to that is not biological meaningful, it's just something to inherit in your first iteration. So as you do this, then you come with something that's sort of the average shape as well as the average intensity. I'm going to go through this a bit as well. So we also saw a very interesting effect. Although our data set is highly anisotropic, 0.3 microns in plane, 100 micron out of plane, we were still, because there was enough data set, we were getting some sort of super resolution effect because we had enough sample in the interim spaces if you have enough mice. So we were starting to be able to see things that you couldn't really easily see on a single brain. We were able to see like the barrels are important, the barrels of the cortex, looking at the barrelets in the sort of at the brainstem area, and also seeing the barreloid in the thalamus. So when we saw this again, it was like, okay, then this is like worth the effort to do a big annotation project on. Okay, so to do this annotation project, big infrastructure came into place. We wanted, our anatomists really wanted to have registered data in order to do the annotation. So as they're working on each data set, they were looking at a gene expression atlas, they were looking at transgenic lines, they were looking at projection data, and this was a while back, like before Neuroglancer and a lot of these online web things. So every data set was turned into a volume, and each volume was overloaded into a viewer, such our anatomist got a multi sort of layer view of the data. And then so they worked on one object one by one. 3D annotation is actually very difficult to keep everything smooth. So peak of the project, there was 1 to 3 different annotators came in to help our senior anatomist to make sure things were smoothed out in a 3D space. All right, so in the paper, there's lots of vignettes of the different components, as well as annotation. I wanted to do something different with the cortex. As I said, at that time, or still now, there's a great interest in the cortex. But because cortex is this bent sheet, it's really hard to tell if the differences you're looking at is because there is a biological difference along the depth, or you're actually looking at the different depth of the brain. You're not tilted, you're not looking at the right tilt. So this is where this method of using a Laplacian was already in play as people were trying to measure cortical thickness. So we adopted the same method as well, just collecting the thickness. We also traced the shortest path along each of these streamlines. So now we have a second coordinate framework, which is sort of orthogonal to the curve that we can use. And this allowed us to actually do really more precise annotation, especially in the cortical areas, because you can project information up to the surface, and that's where the annotation was done, and then the annotation is projected back. Especially in areas such as the visual cortex, when our primary visual area is this sort of bent triangle, it's really hard to see the beginning and end of that triangle if you're looking at coronal slices. But if you project it as a flat slab, then and all the data into the slab, our annotators have better ability to determine the different regions. So this is then trying to validate and see when we have data that is have apical dendrites in that direction. If we follow the streamline, does it get approximately upright? So this is 3 different areas in which of the SIM1 Cree KJ18. So after unbending with the streamline, you get approximate upright apical dendrite. So this was then very interesting, because this allowed us to have sort of a historical 3D data, which is in the whole brain, and the ability to then do cross-analysis to data in which you do a surface view. So this paper comes from Anne Churchland's lab, and like that, because I can recognize the drawing, is then the mapping of wide field imaging data, behavioral data, on top of a surface view of the CCF. So now, because our CCF curve can be sort of rebent into it, you can then correlate, compare data from this modality also back with our sort of projection data as well. This CCF is being used by many different groups. Here is the website from IBL and my friend from lunch. And then this is all their NeuroPixel data. Each of those are NeuroPixel probe inserted into the CCF. And so, so that was basically that. So at that time, when we finished in 2017, the CCF have been widely adopted, like in this community of electrophysiology, but also in the cell type world. So a lot of my picture come from a cell type world, because that's like the path that I've taken. And I know there's a lot of sort of use cases in this room as well. So I apologize I didn't get your use case. But this is then having a look at some of the cell type use cases. So the Brain Initiative has this a program called BICCN, the Brain's Cell Sensors Network. And towards the end of 2021, it had this big data package, which is on the motor cortex, and practically all my slides are showing screenshots and figures from that paper. So where the CCF is starting to come into play is for groups that are doing whole brain morphology. So there's ability to then map morphology from all these different brains into a single framework so you can do comparison. And also the streamline has come into play here where people want to compare data of the apical dendrite going up, and even cells that don't have an apical, but you can still rotate it because you have through the streamlines as well. There were many examples now. Very typically, if you see full brain morphology, they have been registered to the CCF. Another use case is the mouse-like neuron browser, and now within our Institute Neurodynamics, SmartSpray may be another major data set of full brain morphology. Okay, and then moving forward to 2023, there is more data, and with the advent of the spatial transcriptomics data, there's even more sort of need for an anatomical framework. So spatial transcriptomic data allows you to do hundreds, thousands of different gene probe on the same cell. And when used in conjunction with like 10x a cell atlas, you're able to then find a spatial location of each cell type in the brain. So this allowed us to get the 5,000 cell types from the whole mouse brain, map them into the CCF so that you can do spatial distribution for cell types. So not only did our group did it, another group, Xiao and Zhang's data, their multiple Murphy data is also registered as CCF. Different modalities that give you more than like all the genes, the slide seq as well. So and epigenetic data. So we're seeing more and more data being registered in the CCF. I think now the current wave is people wanting to do a data-driven CCF annotation. So I know there's many different projects. This is one I just happened to be involved in with Reza Bassi als in UCSF, in which they take the Murfish data and try to find spatial domain to it. And coming back in full circle, when we first did the Gene Expression Atlas, we also did a version of a data-driven parcellation. So there's 15 years between these two figures. Still looking at the striatum. Okay, and then moving forward beyond mouse, we have more initiative towards doing CCF and spatial transcriptomics for human, macaque, and marmoset, of course not at the same level, and then there's a big development project that's underway as well. And that is also underpinned by a developmental mouse common coordinate framework. This one was led by Young-Soo Kim. We were collaborators, such as also Louise Perez, Jim G, and Zhang Zhang as well. So this is a multimodal template between light sheet and MRI, and there is the primary 8 days, but MRI's data was also collected in between days so that there's a trajectory of transform that can go from one age to another along the whole spectrum. Okay, I am going to move along now. Okay, so again, it's been 10 years, and whenever I give this talk, these are often the questions that I get. Primarily, how do I register my data CCF? Luckily, there's a whole sort of community of algorithm developers that we can redirect people to. But even more interesting, there's a lot of people who are doing things to enhance a common coordinate framework. They're doing more annotations, they're making new templates, and they're also fixing problems that exist. So what we want to do is to think about how do we take all of this into account so that it becomes sort of a bigger community effort to add and enhance to the CCF. So some of the things that people have done already, number one request, where is my Breg stereotaxic coordinates in the CCF? Folks have solved this, especially for the neuropixel component. We want to take that back into a framework so that people can share with a wider audience, because many people have this question. There is the problem with the CCF missing the cerebellum and the olfactory bulb and the spinal cord. So the Blue Brain group with Sebastian led by Henry and Jan Balaji, they have spent actually a year or so filling out the missing pieces and also estimating some laminas, and I think they also annotated the barrels. So we're going to take a look at that to see how we can bring that back into the framework. And importantly, now that we're going to talk about more than one thing, we want to bring different versions, different components from people. We are going to need a sort of a metadata model in order to represent all of these things that we're going to bring together. So this is a call out to this Atom model. There's a Linkermel version of it too, which we're going to talk about next. And to finish off, I'm going to show a pretty picture. We are going to solve some of the problems by putting a portal together, in which case we can document each of these versions and also work with people to come and publish their data asset bit template or annotation, and the ability for people to access it through open data and visualize it using NeuroGlancer. Okay.
Thank you very much. We have to move on towards the trial session. I have a question.
Yes.
So you mentioned marmosets.
Yes.
And you mentioned it's not going to be at the same
Yes.
level. But a lot of tools are being developed to do genetics, all sorts of genomics and sorts of things. They're moving towards marmoset. Why is marmoset not going to as detailed?
I think it's all about like where my interaction with Marmoset would be. We're building a common coordinate framework. It is going to be an MRI-based one. We then are going to pair that with a lot of spatial transcriptomic data as well. So in our world, that is the framework where we land to. But I believe that, as everything else, there's going to be a growing community of people putting data into it. So I think while by ourselves it won't get there, but I think as a community, against with the sharing through this portal, we will have more stuff.
Yeah, hi, Lydia. So at the beginning, you mentioned this kind of super resolution effect you get in two-photon serial sectioning. But do you have kind of measures of the brain-to-brain variability for all these different areas, which would be particularly critical, now that you're trying to drive things like neural pixel probes or other probes, through multiple areas in the brain? Is there a variability map that goes along?
We do have a variability map. It was part of our publication.
Ah.
And again, we didn't share it widely, but we do because we had order transforms as well. So we can regenerate the variability maps. And then probably with putting the rest of the data in, we can probably generate like newer ones too. Yeah.
Okay, thank you.